Okay, we might get started. So welcome to today's signature lecture of the ARC Center of Excellence for Enabling Eco-Efficient Beneficiation of Minerals. I'd like to start by acknowledging the traditional custodians of country throughout Australia and their connection to land, sea, and community. And we pay our respects to their elders, past, present, and emerging, and extend that respect to all Aboriginal and Torres Strait Islanders people today. So today, we're lucky enough to have Professor Marek Pawlik from University of British Columbia, who's there in the evening, maybe about 7 o'clock in the evening. Rob Davis there is about probably similar time. Most of the rest of us, it's around noon. Anyway, we're lucky enough to have Marek talk to us today about flocculant properties, flocculation, and insight into the structure of the flocculated sediments. So I'm looking forward to hearing this. And with that, thank you for joining us, Marek. Yeah.
Thank you very much, Professor Franks, for introductions. I'm thrilled to be here, to be able to speak to you about our research. This is a very humbling invitation, and I also pay respects to the custodians of your land, as we ourselves find ourselves similarly on unceded traditional land owned by the Musqueam people here in Vancouver at UBC. So the title of my presentation is Flocculant Properties, Flocculation, and Insights into Structure of Flocculated Sediments. I am in a mining engineering department, but I am also a professor of mineral processing, and my interests are basically in fine particle processing, surface chemistry, interactions between particles, flocculants, polymers, surfactants, and so on. The work presented today is essentially a result of probably 3 or 4 PhDs, so some of the names that you will see are basically in references, I guess. Those are pretty much my PhD students, or most of them. So briefly about the content, we'll introduce the subject, polymers in mineral processing, applications, uses, and so on. We'll then switch a little bit our attention to polyacrylamide-based industrial flocculants. When it comes to polymer properties, we primarily look at the intrinsic viscosity of polymers, a rarely used parameter in flocculation work, but we find it very useful in terms of understanding how polymers behave in water. Then we'll look at a model system, flocculation of very fine quartz using the same polyacrylamide-based flocculants. And we will look at this relationship between flocculation performance, the intrinsic viscosity of the polymer, and the what we call chain flexibility. This is a very empirical way of looking at it, but we view what we call intrinsic viscosity ratio as a flexibility value. It's essentially a reflection of the ability of a polymer to stretch or coil in solution. We can measure that by following changes in the intrinsic viscosity. We then got ourselves into X-ray computed tomography as a method for characterizing in situ flocculated sediments on a small scale, I must admit. This part of the work was primarily to develop methodology, to develop algorithms. This is a pretty complicated methodology and requires a lot of attention and mathematical evaluation, but the data are extremely useful. So we'll look at pressure filtration of sediments, but we will also look at floc properties, measurements on single flocs, again using X-ray CT in our national Canadian synchrotron facility in Saskatchewan. And we'll finish with a summary of the data. So polymers in mineral processing, they are used everywhere. That's probably not an understatement. They can be used as dispersants, depressants in flow flotation, then as dewatering aids, coagulants, flocculants, and so on. Some of the more unusual applications are as dust suppressants, perhaps at sites where we have to suppress the dust from moving trucks and so on. They are also used under a very wide range of conditions, particularly in terms of ionic strength. Polymers are used in water, in seawater, in a saturated brine, for example, in potash flotation, a process that is carried out from a saturated potassium chloride, sodium chloride brine. Low temperatures and high temperatures, broad variations. In places like northern Canada, the temperature can vary between -40 and +40 centigrades. So that's basically maybe not the full range of temperatures, but certainly it's an environment where you have to pay attention to what happens as a function of temperature. Properties of polymers, pretty broad molecular weights from a few thousand all the way to 10, 20 million or so. Degrees of substitution and anionicity, some of them are natural. Probably the best group that I can think of are polysaccharides. Polyacrylamides, synthetic. This is the group that we will look at in flocculation work, and they are usually soluble in water. And I'm highlighting the word usually because polysaccharides, for example, are not completely soluble in water. They tend to form colloidal dispersion. To dissolve starch, there are methods that describe, using an autoclave at a high pressure and high temperature to force starch to go into solution. Polyacrylamides, for all practical purposes today, we will assume that they are fully soluble in water, although there are papers that may not necessarily agree with me, but again, we don't see any major evidence of solubility here on our data. So for all practical purposes, we will treat them as soluble in water. So again, from there are dozens of formulations. Even polyacrylamide itself can be produced from essentially of any molecular weight. Or any molecular weight distribution also. That type of information is almost never available to the user. You have to bring a sample to the lab and test it yourself to try and figure out what you're dealing with. There are no clear selection guidelines, really. If you were to say we have a certain type of tailings at a given pH produced in seawater, what exactly would we use to flocculate that material? So degree of substitution, similarly, it's very difficult to find what that degree, what that number is. We'll talk a little bit more about it in a minute. They are typically vaguely specified in data sheets as weakly anionic, moderately anionic, maybe highly anionic, but you will never get an actual number, which is something what we looked at as well. So whether it's a dispersant or a flocculant is a function of a molecular weight. Generally speaking, dispersants will be of low molecular weight, and flocculants and coagulants used in dewatering, they will typically be of much higher molecular weight than dispersants. But again, polymers of the same class, depending on the molecular weight, can function as dispersants or as flocculants. So mode of action, adsorption is certainly the fundamental process responsible for what the polymer does. Adsorption can be selective, it can be non-selective. From the dewatering of tailings perspective, we are dealing with non-selective adsorption. All components of the suspension should be flocculated by the adsorbing polymer, but often adsorption is blamed for poor performance, lack of performance, failure essentially in any of these applications. And typically the focus, particularly in mineral processing work, is on the surface properties of particles, things like surface charge, zeta potential, wettability, floatability, rheology of suspensions, and so on. But it's typically very difficult to draw general conclusions because it's very difficult to find studies that will cover a range of samples that give you, if you like, would allow you to draw some general conclusions about how polymer properties affect the results. Especially when you're dealing with samples with possibly different degrees of anionicity, it's important to characterize that, to be able to say, okay, the degree of substitution is 30%, molecular weight is X, therefore the expected results should look as follows. These types of conclusions are very difficult to draw unless you test a bunch of samples with well-characterized properties. So again, little effort is really made to look at the polymer itself. So some of the questions that we were interested in is, well, how do these polymers behave in water? What's the intrinsic viscosity as a function of temperature, pH, ionic strength, and so on? And how does that behavior, that's the connection here, how does that behavior in water affect the flocculation performance of these polymers? So jumping into polyacrylamides now, PAM, pure PAM, should be non-ionic. It's a polymer that doesn't carry any functional groups that can dissociate, that can produce anionic or cationic sites. However, industrial polyacrylamides are deliberately hydrolyzed to actually produce a fraction of anionic groups. Okay, so kind of block structure here of a polymer can be shown as a copolymer of acrylamide and acrylic acid. So acrylamide is on the left, and the acrylic units are shown on the right, depending on the ratio, how many of those. The polymers can be more or less anionic, and then we can define the degree of anionicity of a polymer as a ratio of the number of acrylic units in the chain to the total number of units in the polymer. So it's a simple definition based on, well, this type of diagram. We need to know the number of units of one type, total number of units, and we can define the degree of anionicity or degree of substitution. These are synonymous. They are used interchangeably, and I will probably use them on different occasions in different ways. It's easy to define, but it's not so easy to measure. We developed our own little technique for our PAM samples, based on the observation that acrylic units typically come as sodium salts. So we were not measuring directly acrylic units, we were measuring sodium and then deducing the amount of acrylic, the number of acrylic units based on the concentration of sodium. So assuming that there are no sources of sodium other than the polymer itself, and you can actually take some corrections to, even if there was some sodium from the environment, from the way the sample was prepared and so on. It is actually a pretty robust technique. It's based on total organic carbon analysis of a dilute polymer solution to measure the carbon content, which allows us to calculate the denominator of that definition. We can calculate the total number of units. And if we figure out the amount of sodium, we can actually calculate the number of acrylic units, which will give us the value of N. And with these two numbers, we can then start calculating the degrees of anionicity. That was an older method. And again, the name Arina Itwi, those of you who work with flocculants, actually Isao Arina Itwi is now the manager of R&D with Scienceco, or a parent company from Solvay, which is a parent company from Cytec. So very much in the middle of this flocculant and polymer business. So anyway, we were then able to essentially determine ourselves what the degree of anionicity of our sample was. And then this is what's probably unique about our results, is that this is not a single polymer study. This is a bunch of polymers well characterized. We know the intrinsic viscosity, we will know how they behave in water, and we will then translate that behavior to the flocculant performance. So the intrinsic viscosity, if you're not familiar with the term, and again, this is not something that people commonly do when they work with polymers, it's essentially a measure of size, shape, and conformation of a polymer in solution. It is basically related to the hydrodynamic resistance of a macromolecule to flow. Typically, intrinsic viscosity changes with molecular weight. It increases with molecular weight, with molecular size. And that relationship is given by the Mark-Hoving-Sakurada equation. It's in the middle of a triangle here. So the viscosity in brackets, that's the intrinsic viscosity, and it's a function of molecular weight m. There are two parameters there, K and alpha actually has a physical meaning, shown again in this triangle in the corners. For a compact sphere, alpha should be 0. At the other extreme, if we have a rigid rod, the alpha value will be 2, and most of the polymers that we deal with are in between. So for polyacrylamide, these values, the value of alpha is depending on how it was obtained. It can be between 0.7 and 0.8, so that places us roughly in the bottom left corner of a triangle, indicating that polyacrylamide by nature should be a flexible chain, and that water is a good solvent for the polymer. So again, it goes to the solubility of the polymer and the fact that it should be flexible. A complicating factor for polyacrylamide is that, so these values then were obtained for which degree of anionicity, right? Because if we have a range of those and we can have such a variety of degrees of substitution, then which exactly value is it related to? And there is no straight answer to it. It turns out that these values were obtained for a range of samples using high ionic strength environments, so forcing the polymers to essentially coil in solution, as shown in the diagram on the left. But essentially that alpha value will depend on what conditions the polymer was obtained on. The values were measured in. So the The carboxylic groups, then, if you think about them as negatively charged along the polymer chain, they may result in extension of the chain under favorable conditions. Electrostatic repulsion between these groups will extend the chain. And if the degree of anionicity is low, meaning very few, if any, of these groups are on the polymer chain, there will be no electrostatic repulsion, and polyacrylamide will tend to coil. Even the same polymer, even highly anionic polyacrylamide, depending on the salt concentration, the ionic strength in solution, may go from that extended conformation down to coiled, or vice versa. This is actually a reversible process. If you play with pH back and forth, you can actually see that changing pH will either result in an extended conformation or in a coiled one. Generally speaking, extended conformations, those that will oppose flow, right, they will be of high intrinsic viscosity. And anything coiled will be of relatively low intrinsic viscosity. And again, we were interested not necessarily in the alpha value, we were much more interested in this transition between coiled and extended conformation, and how that ability to do that change in the state of the polymer, how is that related to the flocculation process. So this is our setup. The measurement is based on dilute solution viscometry. So these are extremely dilute solutions, maybe 10, 20 ppm in water. They have to be Newtonian. The definition of dilute is actually given mathematically at the bottom of my slide. The relative viscosity must be less than 2. And the highest viscosity, the highest concentration that can be tested in this measurement cannot exceed a reciprocal value of the intrinsic viscosity. So intrinsic viscosity has units of concentration, volume over concentration. That's just a convention in polymer chemistry. So the concentration then is a reciprocal value of the intrinsic viscosity. So it's a small scale measurement, really. You place a polymer solution in the bulb in this U-tube type Cannon-Fensky capillary. The solution is pumped from the bulb into the right arm, and then pressure is released, and the solution is allowed to flow under gravity, and it passes between, and the time it takes for the solution to flow between two markers, they are barely visible, but they are just above and just below the upper bulb, and the time it takes for the solution to pass is proportional to the viscosity of that solution. You have to perform this measurement for a number of concentrations, and then you can start plotting data as a function of polymer concentration. So the raw results are in the form of kinematic viscosity. The capital K is a capillary constant that allows you to recalculate the time, the flow time to viscosity. That letter V, that's a correction for gravity, which is typically 0 for a flow time on the order of 60 to maybe 100 seconds, which is typically what we aim for. And once you have those raw results, you can then start calculating all sorts of other types of viscosity, starting with relative viscosity, which is the kinematic viscosity divided by the viscosity of the solvent. Then we have specific viscosity, which is relative viscosity minus 1. And finally, we have reduced viscosity, which introduces concentration into the data. So it's specific viscosity divided by the concentration. And then there is a number of equations that allow you to calculate the intrinsic viscosity. The general polynomial equation, it's essentially a polynomial function as different powers of concentration. You can have many terms, typically 3 or 4 are enough to describe the relationship between the reduced viscosity and concentration. A truncated version of it, or an approximation, known as the Huggins equation, is very common for measurements on non-ionic polymers. And you can see that the Huggins equation is essentially, well, the first 2 terms of the general polynomial term. We also found the so-called Fedor's equation to be particularly useful for anionic polymers, because they do not quite give this nice linear relationship that the Huggins equation would suggest. So we found that the Fedor's equation gave us this linearization of the data, but you need to plot a couple of pretty acrobatic quantities here. One is a reciprocal value of the term on the left, and then you have to plot this parameter as a function of reciprocal concentration. You get a straight line, and the slope of that line gives you the intrinsic viscosity. We typically use two of these extrapolation techniques to make sure that we are roughly in the same range when it comes to our readings, typically the general polynomial equation plus either the Huggins equation or the Fedor's equation. And that's how the data typically would look like. So the polymers of interest to us is a group of 5 polymers. They were provided, like I said, by Cytec. And back in the day, we still get samples from Solvay. We used to get samples, sorry, from Solvay. Now we connect with ScienceQuo to get the latest batches, if you like. But that's kind of how the companies evolved. Cytec became Solvay, then they sold the flocculant business, and then it became ScienceQuo. But the 5 polymers are N100, A100, A110, A130, A150. These are our measured degrees of anionicity from 2.5 to about 48%. So in the case of A150, nearly every second unit is basically an acrylic unit, while in the case of N100, this is the least anionic chain. We then realized that the numbers basically indicate what the anionicity is, that, you know, A130 with a 30% anionicity, A150 with 50% anionicity, and so on. But that's how typically the data will look like. The data on the left are for the N100 non-ionic polymer. Like I said, these types of polymers typically give an excellent fit to the Huggins equation, and the intercept of any of these lines with the y-axis, the reduced viscosity scale, gives you the intrinsic viscosity values. Similarly, in the case of an anionic polymer, you can see the 4 anionic polymers here from A100 to A150. They do not follow a linear relationship, and in this case, you need another way of extrapolating the data. So we used the general polynomial function in combination with the Fedor's equation. And again, extrapolating the data to 0 concentration gives us the intrinsic viscosity. So the intercept value, these points here, give you the intrinsic viscosities. So you can do this. You can probably appreciate that you can do this for all sorts of conditions. You can do it as a function of temperature, pH, ionic strength, and so on. And you can look at all sorts of trends as a function of the degree of anionicity of these polymers. So that's kind of a summary of all the data. On the left, we have intrinsic viscosities of these 5 flocculants in different pH values, different temperatures in distilled water. So first thing to note is there is really no effect of temperature, which is great, because we didn't need to worry about what is happening in our lab when we are taking these measurements, even though you can do this in a water bath, but you prepare samples maybe in some other environments and you end up with unreliable data. In distilled water, there seems to be an effect of pH. The numbers decrease quite dramatically from the data at pH 5.7 to pH 10.5. But this is an experimental artifact here because remember that in order to go from pH 5 to pH 10.5 or so, you need to introduce sodium hydroxide, quite a bit of it. So you are changing the ionic strength. So what you see here is a combination of the ionic strength and pH, right? Those two effects together on the intrinsic viscosity in distilled water. Once you introduce constant ionic strength using sodium chloride, the effect of pH disappears, right? Now, in this case, all the polymers, basically, all the anionic groups on these polymers are screened by sodium hydroxide, by sodium cations from sodium chloride, and effectively the polymers coil and they give you very similar intrinsic viscosities. So this is actually a typical effect of an electrolyte, okay? Any electrolyte will do this to a charged anionic polymer. Okay, typically you observe a decrease in the intrinsic viscosity related to the fact that the charged groups on the polymer are screened by the cations or anions from the electrolyte and the polymer coils. So these were the intrinsic viscosity data. Our flocculation work was done at pH 5.7. So at some point we will come back to these results, but only collected at pH 5.7. So moving on quickly to flocculation. Dilute quartz suspensions, pH, like I said, 5.7, various dosages. The quartz sample had an average particle size of about 3 microns. We arbitrarily chose different flocculation conditions. When you submit papers on flocculation, you are often asked, well, what's the shear rate, what's the power to volume ratio, all the hydrodynamic conditions, and so on. In the first scenario, in the first method, we can actually report that because we know the power draw, we know the gap between the baffle and the impeller. But in the other cases, we decided to be careless, all right? Like we were handshaking in a vial, you know, very difficult to reproduce, really. Depends on who does the test and how much attention they pay to it. We also had a sort of like a rotating arm kind of device where you place your settling column and you rotate it X number of times. You can control the RPMs manually or electronically if you want. It was recommended to us by one of our sponsors. So under these three sets of conditions, we did all this flocculation work. We also followed the changes in the turbidity of the supernatant. After the solid settled, we measured what was left in the liquid above the sediment. And we also measured the amount of polymer in solution. So knowing the amount of polymer in solution and knowing how much we added, we were able to calculate the adsorption of the polymer on the solids under flocculation conditions. So that's what the results look like. Whether we used cylinders, whether we used handshaking or inversion, the data fall on very similar trends. You can appreciate that the absolute numbers for the initial settling rate and turbidity values are different. That's a reflection of different hydrodynamic conditions. But the trends, as a function of the polymer type, in this case, we plot everything as a function of a degree of anionicity, the trends are nearly identical. We always get this maximum for A100, the weakly anionic polyacrylamide. We always get very poor performance from the non-ionic polymer, N100, and then we get these sort of not so great results for the other anionic flocculants. So our conclusion based on this would be that A100 is basically the best polymer for flocculation of quartz at pH, I guess, 5.5. It gives us the highest settling rate and it gives us the lowest turbidity for all the conditions. And you can even see that some of the more anionic types, they actually give pretty high turbidity values compared even to the non-ionic type. So that means that, you know, we were not really succeeding in flocculating quartz using these most anionic types. So these two pieces of information together tell us something about which of these are the best. So that's kind of a guideline for flocculation of fine silica and fine quartz. You need a weakly anionic polymer because anything less or more anionic will basically give you inferior results. These changes are not a result of changes in adsorption because we had the adsorption data shown in the bottom right graph. And we found that when the data for flocculation were collected, the adsorption density of all these polymers was nearly the same. And it was practically complete, right? All the polymer added to solution was essentially adsorbed on the solids, all of these polymers. And yet all these polymers gave us quite different flocculation results. So this was flocculation. And then we decided to look at the polymer and correlate properties of the polymers with the flocculation data. As I mentioned, we defined the intrinsic viscosity ratio here as the intrinsic viscosity value in distilled water at pH 5.7 divided by the intrinsic viscosity value in sodium chloride. So the value in distilled water can be viewed as a value for the extended chain, while the value obtained in sodium chloride is a result of polymer coiling, and it's essentially a value for a compact sphere or a polymer in a very coiled, highly coiled configuration. So we looked at that ratio as the ability of a polymer to go from one state to the other, right? How flexible are they? Can they really conform to one shape? Can they really uncoil? Can they coil back and so on? So the highest intrinsic viscosity ratio was again found for the A100 polymer, and the data, the intrinsic viscosity ratios for these polymers, are plotted now together with the initial settling rate. And interestingly enough, the two sets of data, you know, polymer behavior in water, and then the flocculation power of the polymer towards fine silica, the two sets of data essentially overlap. The polymer that has the highest intrinsic viscosity ratio, in other words, it's most able to stretch and coil in solution. We could think of it as most flexible. This is also the most effective flocculant. And our explanation for it is you need that level of flexibility not only to bridge particles, but also to conform to the size and shape of flocks. If you have a rigid rod, its ability to really form compact, you know, or competent flocks is not so great because it will not be able to really, again, to conform to the size and shape and, the dynamic nature of flocks. If we have spheres, well, they will absorb densely, right? So N100 is one of those. But the ability to bridge particles is quite limited because they don't have the reach, if you like, because of the molecular compacted nature. They do not have the ability to bridge many particles at the same time. So when it comes to so that's basically probably the most important finding for us, that for the first time we are able to say that the flocculation result is related to the flexibility of the polymer, not only to the molecular weight, not only to chemical conditions in the water, but also to the nature of the polymer itself. And again, flexibility in our view is really a key parameter when it comes to using polyacrylamide for flocculation of fine silica. So going back to that sort of physical explanation for it, we're flipping between two conformations, extended and coiled. And again, this ability to go between these two states can be used as a measure of polymer flexibility. And again, this flexibility seems to promote flocculation and fast settling. So the absolute size, the absolute molecular weight is not really that important here. It's actually the conformation in solution and the ability to adapt to different chemical conditions in water. This is what defines the flocculation power of polyacrylamides. And that's kind of related to what we know about what polymers do on mineral surfaces. This is a very well-known figure from a book by Robert Hunter, some time ago, showing configurations or conformations of adsorbed polymers on solid surfaces. So it's quite easy to imagine that the rigid rod will never be able to assume all these conformations. It will probably not produce loops. There may be some tails dangling from one end or another, but there will be very limited ability to bridge multiple particles. If we have spheres, well, again, there will be no tails, no loops. These are the conformations that are responsible for flocculation. And again, you would not expect a great performance from that type of polymer. So on one extreme, rigid rods, that's basically the most anionic PAM. It's rigid because the electrostatic repulsion between the groups keeps the polymer stretched, right? Any collapse on its own and intramolecular bonding is very difficult because that would need to go against electrostatic repulsion. So in this case, the polymer is effectively rigid and not quite powerful as a flocculant. On the other extreme, the most non-ionic polymer, N100, again, it's completely coiled. The intrinsic viscosity is very low. It doesn't uncoil. It is unable to bridge particles into flocks. So the weakly anionic PAM, the A100 variety, this is the one that offers the best compromise between these two extremes. It's most flexible. It is able to bridge particles. It is able to conform to the size and shape of the flocks. So that was probably the most important finding from all the work that we did on polyacrylamide. We have some data on minerals like kaolinite, alumina, and so on. So depending on conditions, this still holds, but it's not necessarily universal. So I would add here another condition, but this is probably true for polymers adsorbing by hydrogen bonding, because it's very difficult to imagine that polyacrylamide would adsorb through any other mechanism onto silica other than hydrogen bonding, right? The silica is negatively charged typically under these conditions. The polymer itself is anionic, so electrostatic repulsion dominates. So at best, you are looking at adsorption by hydrogen bonding. So that sort of brings us now a little bit to flocculation and characterizing sediments. So we often do, settling tests in the schematic shown on the right. That's basically what we did with flocculation of quartz. We can measure the quality of the supernata. We can use all sorts of optical methods to determine the settling rates, turbidity, and so on. And then the whole trick is to translate these results to some kind of operation of a large-scale thickener. But the transition or the thought process here takes a bit of a shortcut, if you like, and because there is a tendency in publications to say that high settling rates, large flocks, high turbidity, very low turbidity, and so on. But all of these mean automatically that we will have very easy way with pressure filtration, with further mechanical dewatering of these sediments. So we decided to look at that correlation, if you like, and this interface between what happens during settling and thickening and then pressure filtration. So to do that, we decided to turn to computed tomography, X-ray computed tomography, as a way of monitoring what is happening in flocculated sediments. We will look at some results obtained this time, unfortunately, for a cationic PAM, because we were focused more on developing algorithms and trying to figure out what the technique can tell us. But still, some observations turned out to be quite interesting, even though we basically used a different type of polymer. So this analogy, that high settling rate in a batch experiment in a graduated cylinder, that it's equal to easy filtration. This is not exactly what will be the case in the data that we will look at. So, characterizing sediments using X-ray computed tomography. You are probably familiar with the term CT scanning, right? It's essentially a medical way of. It's a medical method for looking at broken bones and all sorts of 3D imaging. There are some really interesting papers on this. I couldn't resist showing the picture on the right. It comes from a paper on leaching columns, on leaching in a column, I should say, and then following changes in the structure of that column of rocks using a medical scanner. So in this case, instead of a human body, you can see essentially, a pile of rocks sliding in, and then they had all sorts of very interesting data coming out from the scanner. But in terms of principles of the measurement, you have basically two ways of taking CT data. In a medical scanner, the sample, right, the column of rocks here, is stationary, and there is an array of detectors around it. So the sample can be scanned from all these different directions, or irradiated in a sense. And so the sample is stationary, and you have an array of X-ray sources basically shooting at the sample. This is very difficult. This can be easy. This can be difficult. But in micro CT, in microcomputed tomography, the situation is kind of reversed. The source of X-rays, as shown by this box on the left, is stationary, but the sample is now rotating. So that gives you all the 3D data that you would like to know. You need a computer to do this, really, because you will be looking at which point X-rays come through the same point, what type of information you can get from this direction, But the principle is essentially the sample is rotating while the X-ray source is stationary. The physics is governed by the Beer-Lambert law. This is a typical light absorption law used in UV visible spectroscopies. In this case, we have X-ray absorption. The parameter that is of interest to us here is the linear attenuation coefficient. It's a physical property of a physical constant for a given material. And then everything else is pretty much what you would expect. Path length, beam intensity, the density of the material, and then you can collect all that information as a function of position in the sample. We built our own little filtration apparatus. So this is a microfiltration unit, and the idea was to perform a compression test and then transfer the cell to a medical scanner. I mean, we had access to a unit in dentistry, so we were basically bringing tailing samples to a clean lab in the Department of Dentistry at UBC. But the idea was to perform a filtration test, and without removing the sample, moving it now to the whole setup was then moved to the CT scanner. So you can see a picture of a flocculated sediment on the left, and then the cell itself seen in the CT scanner. We were interested in 4 positions just above the bottom, roughly 1/3 of the way, 2/3 of the way, and near the top. The scanner, the voxel size, or in this case, it's a 3D unit for resolution, is about 7.4 microns. And again, cationic flocculant from Cytec. So micro CT, the raw results from CT scanning are unattractive, I would say. It's a grayscale image. There is really not much to do until you start mathematically playing with the numbers. So in our case, we used what's known as a random walk algorithm to distinguish between liquid and aggregate phases. And we define aggregates as consisting, or flocks, as consisting of solids and water. Once you do that, you can then, with proper calibration of the grayscale, because that's another aspect, to be able to tell how much of silica do I have in a given voxel or in a given volume of suspension. I have to know that I am really looking at silica. So we calibrated this type of signal using, well, deionized water at one extreme, quartz crystal at the other, and then polymer stabilized silica suspensions in between. So we deliberately, again, overdosed the polymer, actually one of the same flocculants that I mentioned earlier. So once you overdose a polymer, you have a sterically stabilized suspension. The material doesn't settle because that's a concern in the case of dilute or more concentrated suspensions. But in a sterically stabilized suspension, the settling is very, very slow. So we were able to add a few points on the line. We had our relationship between the CT number from the Beer-Lambert law, and then we could correlate that with the mass density. And then we added colors finally, right? So this is post-processing. And this was probably the first image where I got to see the actual densities of flux, density distributions. What we found interesting here is that the highest recorded local density was about 1.8 grams per cubic centimeter, so quite far from the density of pure quartz. But you can get a sense of how the numbers, and this is a single slice, and you have hundreds of thousands of those, and you can start producing 3D images as well. So we measured floc densities. From this, you can look at solids contents. We found quite interesting that many flocs showed, if you like, centers of high density, and then they were going fluffy, if you like, all around. So those were properties of individual flocs. Well, we'll talk about that in a minute. But that's basically how you can manipulate the results and what type of information you get from it. And again, you can. This is a single slice. You can do it in 3D. So looking at a sediment structure in 3D with this technique, once you calibrate the signal, becomes quite informative about what is happening inside the sample. So these are some of our filtration results. So again, microfiltration in that cell, and then we moved the cell after a step filtration test to the micro-CT apparatus. So again, just to define a few terms, aggregates in our discussion, these are consisting of quartz and water. So we step pressure, right? We applied pressure until we couldn't really get anything out of a sample. So when you do that, when this is done at low pressures, you can see that most of the changes really take place at lower pressures. We have three graphs here, one showing solids volume fraction in the sediment. So that's what we typically call the solids content. Then we have aggregate solids volume fraction, the concentration of solids inside the aggregates. And then we had aggregate volume fraction, meaning how much of the space is actually occupied by flocs, right? So not just solids, but entire flocs. So what happens here is that at lower pressures, most of the changes in these graphs occur basically between between 0 and 3 pascals, and most of the changes take place in the aggregate volume fraction. So what that means is that when we, at lower pressures, we only expel pore water from the sediment. The sediments themselves are not really changing. They rearrange, they may pack more densely. We remove most of the pore water, but the sediments are not impacted. And you can see that the aggregate solids content for those red and green lines, so those two sets of data reflect two measurements. But there is really no change in the solids content inside the aggregates. We removed water from between aggregates, but we did not really destroy the aggregates. Only when we go to much higher pressures to show an extreme value, 75 kilopascals, you can see that basically the entire volume is now occupied by aggregates. The solids content in the aggregate phase is the same as the solids content in the sediment, roughly 45, maybe 50% by volume. So that means that finally, at very high pressures, we started to collapse the aggregates themselves. So at low pressures, filtration is due to expulsion of pore water. It doesn't really destroy the flux. It doesn't really expel water from inside the aggregates, and only when you go to the extreme pressures, that's when finally flux themselves start collapsing. But before we really collapse the structure, this is perhaps an opportunity to think how we run filtration. Maybe stepped pressure increases, gradual pressure increases to maximize expulsion of the pore water. that could perhaps be beneficial in early stages, because once we go to very high pressures, we basically collapse the entire sediment and the experiment basically stops. So using now the same technique, but this time for properties of individual flocks. This work was done at our synchrotron facility in Saskatoon. We were granted 3 days of beam time, so you need to send people as a team. They work 24 hours a day for 3 days non-stop. Some parameters of the facility. So again, the same flocculation, the same flocculant, sorry, the same solids. In this case, after flocculation, we transferred the flocks into an agarose solution for setting, and because we found that this is the best way to preserve. This is actually described in publications of many studies that did similar work on flocks aggregates. Agarose basically preserves the size and shape of the flocks, and it also allows you to use maybe other techniques, confocal microscopy, photography, whatever you prefer to look at the flocks up close. And for this we prepared 3 different batches by simply pouring different amounts of flocks into the agarose solution. So we had 3 subsamples with a total of about 10,000 flocks. We counted those again with the CT scanner. These are the distributions again from the CT scanner, size distributions, cumulative. So different samples, slight variations in the frequency, but the cumulative curves were the same. So that told us that the samples were quite reproducible of the entire population that was used in these experiments. And then we started playing with the data. Okay, so we were interested in knowing, for example, what is the structure of flocks? How do they grow? How does flock growth proceed? Is it a result of literally flock growing bigger and bigger, or is it a result of aggregation of smaller flocks? So the plot on the left provides some insight into this type of mechanism. So the 1-to-1 line drawn through it basically means that those are single flocks. We call smaller flocks, we call subunits. So basically one subunit could be a single flock, but it could be a building block of a bigger flock. So any flock along this 1-to-1 line would be basically a single flock of a given volume. And there is actually quite a significant population of flocks that are essentially single units. They vary in size, but every flock to the right of that line is essentially composed of subunits. Okay, and then you can have more, and so this is considering the frequency scale here. This is in fact a 3D graph with a maximum roughly around the number 2 here. This number 2 highlighted in sort of surrounded by the light blue circle. But basically along this line we have single flocks. We then get to a point where we still have a small population of very large flocks, but they are entirely composed of smaller subunits. We were also able from the data to plot the distributions of single subunit flocks, right? Those that consist only of single clearly visible units. We can tell whether it's a single unit or subunit by following changes in the density around the flock. If you can find areas or planes where you have lower density consistently between 2 areas, it's a very good sign that you have 2 different flocks coming close into contact. If there is no change in the density, then very likely flocks came together as a result that this flock basically grew as a result of adding more solids onto the core. So we have 3 distributions here, distribution of single flocks, distribution of subunits that we found in multiple flocks, and then distribution of these very large flocks composed of multiple subunits. So going all the way to the top right, there is a small population of flocks in the top right corner that they always consist of very large subunits. They do not exist as single flocks. In other words, these largest flocks had to form as a result of aggregation between smaller flocks. We call that type 3 growth. So basically they are so large that they can only form from smaller flocks. In area to the left, where we say type 2 or type 3, so type 2 would basically be growth of a single flock by adding more solids around a core without really attaching smaller subunits. But there is also evidence that we are dealing with growth of much smaller of aggregation of smaller flocks. So we still deal with type 3 growth in that space. Type 1 would be coagulation of individual particles, but because of the size of this material, which was only 2 or 3 microns, and the resolution, the voxel size was about 4 microns, we couldn't quite see those, so they were eliminated. But that's basically, those were not flocks. Those were primary particles aggregating, for example, as a result of changes in pH. So what we find here, what's interesting is that very large flocks form only as a result of aggregation of smaller flocks. Flocks of intermediate size are typically aggregates of much smaller flocks, but they can also exist as single very large units. And statistically speaking, again, these very large flocks from the top right corner, they were only represented maybe 6% or so of the population by number, but they were about 45–50% by volume. So with that information, again, the structure of single flocks, and again, so these distributions again suggest that very large flocks are unlikely to form as a result of growth. They are much more likely to form as a result of entanglements, mechanical entanglements between smaller flocks. And that's how some of these pictures look like. There is a ton of data in these types of images. You can see how the segmentation into subunits went. You can calculate all sorts of parameters for it, including fractal dimension if you wanted to. One particular parameter that we found interesting was the value of A, which is basically the average area of contact between subunits. We felt that this is somehow related to flock strength, because the larger the contact area, the stronger the flock forming from aggregation of subunits. So we did some statistical analysis of how that changes with flock size, flock volume. So not surprisingly, the total contact area between subunits in flocks increases with flock volume. But the average contact area seems to be reaching some kind of plateau. This is no longer a straight up relationship, the graph on the right. There seems to be a maximum average contact area, so meaning that even for very large flocks, their strength is not necessarily increasing. With the average contact value being fixed, very large flocks are probably weaker, which is in agreement with what typically studies show, but very large flocks are not necessarily stronger. So again, assuming that the strength of flocks is proportional to that shared area between subunits, and because there is a change in the mechanism of formation, growth versus attachment, these very large flocks are not necessarily stronger or more resistant to filtration when smaller subunits. So again, in relationship to the compression data, jumped 2 slides. So at low applied pressures, as I mentioned, when the majority of filtration is by expulsion of the interpore water from the flocculant phase, there is a change in the aggregate phase concentration, but that's basically a result of rearrangement of those units. It's not necessarily compaction of flocks, it's simply a breakdown of larger flocks into subunits. This is again in agreement with the theory how flocks form, that the very large flocks, macro flocks if you like, when they are generally very weak, they seem to be forming by mechanical entanglements rather than by surface forces, by polymer-induced adsorption. These are mechanical entanglements rather than, like I said, much stronger surface forces. And when the behavior of very large flocks, again, this rearrangement of subunits seems to dominate filtration at lower pressures. This is where these large flocks kind of break apart, units rearrange, and we only see expulsion of interpore water. Only when you go to very high pressures, you can start now expelling water from inside the smaller subunits. So, and again, later stages, once you go down to those very small subunits, type 2, type 1, that's when filtration becomes very difficult, because it's more and more difficult to expel water from these much stronger, smaller subunits, whether they are small flocks or whether they are large coagula. That remains to be seen, but at that point, basically filtration becomes very, very difficult. So if we could somehow preserve the structure of that sediment that we see at least initially, so again, the presence of very large flocks, this is not necessarily good news because those flocks will break very easily. So perhaps working with smaller flocks, maybe there is a compromise to be made. Let's not target the highest settling rate, meaning we have highest, largest flocks. Let's target something in between, because those smaller flocks are very likely much more resistant to filtration than those very large flocks that we saw in our population. So we're almost up to 1 hour, so just some summary of the data. We find the dilute solution viscometer is extremely useful in collecting valuable data about the polymers, which allow us to draw new insights into what is really important in flocculation. This term that we call flexibility, the parameter that tells us whether the polymers are able to coil or stretch in solution. We see this as a very key parameter in defining the flocculating power of a polymer. That seems to be related to the ability of these polymers to adjust, conform to the size, shape, maybe texture of surfaces among the flocculated particles. So this connection between, again, settling, high settling means excellent performance, deserves continued research. Like I said, we are not convinced that that's really the case, because fast settling of large flocks does not necessarily imply that that those flocks will resist filtration. In fact, we see that they go first, even under relatively low pressure. 75 kPa is not that high. 250 is probably more likely that you encounter in pressure filtration. So, but even in that range, it's clearly visible that very large flocks collapse first, and then the rest is defined basically by the behavior of smaller subunit flocks. So the X-ray data, X-ray CT suggests that these very large flocks, they form by entanglement of smaller flocks. They undergo this early rearrangement into subunits. And then those subunits are in fact much more resistant to compression and withstand pressure filtration much better than large flocks. And again, these are much more likely to be held together by stronger surface forces compared to, let's say, particles held by mechanical entanglements. The CT is this is an extremely powerful technique. The volume of data is incredible. The numbers, the way in which you can statistically evaluate everything, that may take forever, and we are still sifting through some of these results. And we are going back to flocculation with those polyacrylamides and trying to see what is now the correlation between the sediment structure, as shown here using these methods, and again, the degree of anionicity of the polymers. But this is a very promising technique, certainly highly valuable at lab scale. The compromise here is the time it takes to collect the data and the resolution, because it's small voxel sizes on the order of 1, 2, 3 microns, and there are now even higher resolution types. It will take days to collect the same volume of data as it takes at a voxel size of about 7 microns. So, which is some of the dental scanners that we used in our work. But anyway, it's a very interesting technique, very promising, and we found it a lot of fun. We have examples of how you can fly through flocks. You can produce a computer game out of these types of results, where you can take the reader on a path through, these channels and pores and empty spaces. So that really concludes my presentation. I think I'm maybe 1 or